My grandmother had late-stage Alzheimer's before she passed, and my other grandmother had Parkinson's. So being able to study how the brain responds to aging and how we can tailor that response to better deal with the issues of aging, I think is always going to be a very exciting project.
Alzheimer's is really a research area that's in need of new ideas. A lot of ideas that people have tried in the past haven't panned out.
Having an outsider's perspective does allow you to think about a problem in different ways. I started out being interested in leukemia, which is a cancer of the blood. We discovered that we could identify the very first mutations that are in the process of becoming a blood cancer. The same mutations, they also have effects on mature immune cells. And we also know that alterations in the way that these immune cells function can predispose to Alzheimer's disease. But to our surprise, we actually found that individuals who had mutations actually had a decreased risk of developing Alzheimer's. And so this is how we got from studying blood to studying the brain.
What Sid's team had really done is find this human genetic association between the presence of these mutations in the blood and lack of Alzheimer's. Then what they were trying to do is understand whether those mutations could actually be found in the brain and which cell in the brain they might be in.
There are a number of brain banks around the country that have consented people to donate their brains after they die. And so we collaborated with one of these cohorts to obtain these posthumously obtained brain tissue samples.
Each different cell type, in the huge diversity of cell types that make up the brain, has a distinct fingerprint. We realized that most of the mutations must be in the microglia, this brain-resident immune cell type.
The current dogma states that microglia get to your brain when you are 10 weeks old and then are an isolated population. But we've shown that microglia have mutations that they could only have if they are replenished from other cells as you age. Being able to rewrite how we think about a fundamental process is always really cool.
We would like to know what is different about the microglia when they have one of these mutations. Are they perhaps better at surviving into old age? Are they perhaps less likely to stop dividing?
If we can understand what they might be doing to protect the aging brain, we might be able to leverage that therapeutically. The funding from the Knight Initiative for Brain Resilience has really been game-changing for our research and allowed us to pursue these ambitious questions.
This is a very, very new finding, and I think has caught the world by surprise. And being able to interact with the other members of the community, the other researchers in the Knight Initiative, has been really helpful in terms of stimulating our thinking.
Being a part of this whole movement and initiative, it feels really special. It feels like you're a part of something bigger than yourself.